must say it's a great pleasure to start this with beautiful classical music in these really complicated times. So that's actually fantastic. Well, welcome to our first virtual Q&A. We've been doing a lot of Zoom meetings and a lot of WebEx, but never a virtual Q&A. So this is the time to get feedback. We want to thank all of those that participated and were interested. There were a lot of questions. The purpose of this Q&A is to hear your questions and to get more feedback. We want to thank all those that work with us. We're deeply appreciative, and I want to thank all the Friends team who worked so hard on this, Jeff Allen, Diana, Mark, and everyone that was part of it. And now I turn it over to Friends President and CEO, Jeff Allen.
Great, thanks so much, Ellen. Good afternoon, everyone. I'm Jeff Allen, President and CEO of Friends of Cancer Research. Thanks so much for joining us today for our follow-up meeting. As Ellen mentioned, we were really excited by the amount of feedback and the detailed questions that we received based on the August 11th initial meeting. And as we all adapt to new ways of working, it struck me that these are the casual discussions that typically occur in between sessions or in the conference hallways. They provide the opportunity to answer questions and creatively brainstorm together. So that's what we're going to aim for today. It'll be a little different than our past virtual meetings, so thanks for coming along as we try something new. The presentations will be shorter, and since most of you attended the original data release, and the majority of our time today will be driven by your questions. And I'm excited that we have a virtual roundtable of experts to help us out with those and lend their perspectives as well. So to kick us off with a quick review of the ctMoniTR Step 1 key findings, I want to introduce Katie Nishimura, a biostatistician at Cancer Research and Biostatistics, or CRAB, who was instrumental in conducting and reviewing the statistical analyses. Katie, thanks to you and your colleagues so much for your support. I'm pleased to turn the presentation over to you.
Thank you, Jeff. Okay, so let's get started. So as you can see here, a lot of people contributed their time and data to this project. The results I'm about to show you would not have been possible without the guidance from everyone on this slide. The ctMoniTR working group determined the criteria for step 1, which is summarized here. The study was limited to patients with advanced non-small cell lung cancer who were treated with a PD-L1 checkpoint inhibitor. The chart on the right shows the sample size for the final data set, which includes data from 5 sponsors for a total of 200 patients. We used 3 different approaches to quantify changes in ctDNA. All of these used variant allele frequencies, or VAFs, from 2 different ctDNA samples. The baseline sample was collected no more than 14 days prior to the start of treatment, and the T1 sample was collected no later than 70 days after treatment started. The mean, median, and maximum VAF from each baseline and T1 sample was determined, and then the percent change between those 2 samples was calculated. In the blue box, the continuous variable was just the percent change of ctDNA from baseline to T1. In the green box, the 2-level variable was defined based on an optimal cut point analysis, which identified a 50% decrease in ctDNA as the best cut point to maximize differences in overall survival. Finally, in the yellow box, the 3-level variable was created to investigate trends in ctDNA changes, as well as to take into account cohort-specific differences in ctDNA results. I'll now review a few of the main findings. For the 3-level ctDNA variable, we observed strong associations with both overall survival on the left and progression-free survival on the right, with a clear trend across all 3 levels. Patients with a strong decrease in max VAF, the dark blue curve, had the best survival, while patients who had a strong increase in their max VAF, the red curve, had the worst survival. And patients in the intermediate group, the green curve, were somewhere in the middle. Similarly strong findings were seen in the 2-level ctDNA variable, where patients who had a strong decrease in ctDNA had better OS and PFS. Multivariate Cox models show that the 3-level ctDNA variable remains strongly associated with OS, the figure on the left, and PFS, the figure on the right, even after adjusting for other clinical covariates. The orange box in the last row shows the hazard ratio for the 3-level max VAF ctDNA variable. The hazard ratio greater than 1 means that there's an increased risk of an event with each increasing level of this variable, from strong decrease to intermediate to strong increase. In other words, the risk of death or disease progression increases in patients who have increasing amounts of ctDNA over time. Finally, when using the 2-level MATS VAF ctDNA variable, we observed a strong decrease in ctDNA. Strong decreases in ctDNA were also associated with improved patient outcomes, defined here as achieving partial response or better. The downward sloping lines for each cohort shows that the predicted probability of achieving PR or better declines in patients who had an increase or a weak decrease in ctDNA. Logistic regression results had an odds ratio less than 1, which meant that the odds of achieving PR or better was reduced in patients who failed to have a strong decrease in ctDNA. Despite the challenges of working with retrospective data, we were very happy to see a consistent signal across multiple analyses. However, there are many additional questions we would have liked to have asked and that we could potentially answer in step 2 if we collect the right data. For example, how early can a signal be detected? Are similar signals present in other cancers or other stages of disease or other treatments? And how consistent is the signal when looking at different tumor and patient subgroups? Our ability to answer these questions will depend on the amount and type of data that we receive. Step 2 of this project is currently looking for partners who have ongoing trials that have collected data consistent with our recommended collection schedule, or are planning trials and anticipate having data by 2022. One of the biggest challenges from step 1 that we hope to overcome in step 2 is to increase the consistency in ctDNA sampling. This is necessary if we want to answer some very targeted questions. For example, whether ctDNA signals can detect changes in the tumor earlier and more reliably than imaging. This table was developed as part of a white paper presented at the Friends of Cancer Research annual meeting in 2018 and provides some best practices for ctDNA collection. We'd like to propose something similar for the incoming data in step 2. These recommendations are just a starting point to help us align the data that will be collected. Since we're just starting to plan for step 2, we're asking for your help in designing a study that is both analytically robust and clinically meaningful. Thank you very much for your attention and your interest in our work.
Great, thanks so much, Katie. We really appreciate the quick review of the findings. As I mentioned earlier, today we're going to try and conduct this as a virtual roundtable, and we have a great lineup of experts to answer some of the questions that you asked when we presented these findings during our last virtual meeting. I'd like to turn things over to our moderator for today's discussion, Dr. Naveen Zarifa, principal and founder of the NMD group, who's been an instrumental advisor and architect of ctMoniTR. And please remember, as our discussion gets underway, to feel free to submit your questions through the Q&A tool at the bottom of your screen. Naveen?
Hi there, and thank you. And apologies if you hear some noise in the background. There's some emergency construction going on where I am right now. So a little bit like the hallways of ASCO, where there might be others coming through while we talk. So we have a wonderful panel with us today. A number were involved directly with step 1. A number are fresh eyes and will keep us honest and structured as we look to step 2. I will go ahead and introduce the panelists one by one, and then we'll get started on questions. So please keep them coming through the Q&A pane in front of you. So first is John Baden, who's a director of pharmacodiagnostics and precision medicine at Bristol Myers Squibb. Welcome, John.
Thank you.
Next is Carl Barrett, who is the VP of Oncology Translational Medicine at AstraZeneca. Hi, Carl.
Hey, Naveen.
Next, Erik Bloomquist, mathematical statistician at the US FDA and new to the project, so he is one of those fresh eyes.
All right, thank you. Thank you.
Hi, Erik, welcome.
Hi.
Next is Darya Chudova, who is Senior VP of Technology at Guardant Health. Hi, Darya.
Good morning.
Next, we have Matt Hellman, who is a medical oncologist at Memorial Sloan Kettering. Also a fresh set of eyes for us. Welcome, Matt. Okay, and I think Matt will be joining us for just over half an hour. Hi, Matt, welcome. Next is Roy Herbst, also new to the project, though he was part of our panel last time. Welcome, Roy. Roy is a professor of medicine and oncology, director of the Medical Treatment Center for Oncology at Yale. Welcome. Next, we have Antje Hoering, who is the president and CEO of the Cancer Research and Biostatistics Group, who undertook all of this work that you've seen. Welcome, Antje.
Thank you, Naveen.
Next is Geoffrey Oxnard. You know, Geoffrey has been involved with ctDNA for some time. He is currently at Foundation Medicine, where he is VP and Global Medical Lead there. Welcome, Jeff.
Hi, thanks for having me.
Next, we have Rena Phillip, who is Director of the Division of Molecular Genetics and Pathology at CDRH at the US FDA. Hi, Rena, and welcome. Then we have Diana Instrumental Marino Vega, who is the Director of Research Partnerships at Friends of Cancer Research, and definitely the steady hand at the helm of the ship. Welcome, Diana.
Thank you, Naveen. Hi, everyone.
And we also have Chi Shah, who is a Director of Biostatistics at Genentech. Hi, Chi, and welcome.
Thanks, Naveen.
So here you have it, a group of clinicians, of researchers, of diagnostic developers, of drug developers, statisticians, and regulators, all here to answer questions that have come up. And I thought what we would do is pick up on some of the questions we heard last time, just to have complete clarity on what was done in step one. So first, we're talking about ctDNA metrics. And one of the questions that has come up over and over is whether or not our summarization of ctDNA was focused on the variance at baseline, or how did we handle different variants at different time points? So, Chi, I think I'd like to invite you to comment first.
Thanks, Naveen. Yeah, actually, that's a good question, except that the answer is no, because the analysis were actually not based on specific variants, partly due to the limitations of the data submitted by the sponsors. At the end of the day, the analysis considered all variants at each time point. So the mean, median, and max you've seen from Katie's slides are derived from all variants at each time point, and the variance metrics are actually derived from those values there.
Very good. Thank you, Sean. So I wonder if we think about going ahead to step two, Carl, maybe your commentary on this question of variance in ctDNA and time points.
Yeah,
And
he gave obviously the correct answer. I mean, it's important that we consider that these are markers of tumor-derived mutations, and that's why we call them variants. There is always a confounding factor of looking for chip mutations, and each of the vendors have various ways of accounting for that, as we discussed in our face-to-face. But I think it's fair to say that we're really measuring markers of the tumor and how they change with treatment.
Good. And I'm wondering if anyone else wants to chime in on this one. Okay, so
I just want to point out the other confusing factor is germline variants, right? And so you could mistakenly track a germline variant that doesn't move on therapy. And so you both need to find a strategy to eliminate confusing germline variants and confusing clonal hematopoiesis variants, white cell variants, and make sure you're tracking the tumor variants. And that is different depending on assays, but it's certainly an important measure we need to consider as we go forward.
And that's perhaps the challenge of signal to noise that we need to be aware of as we go into step two. So thank you for that. Let's carry on. We're getting some questions through the Q&A. There's one that came up, it's come up today and it's come up previously, and it has to do with understanding what this 50% cutoff was exactly. So I wonder, Antje, if you might tackle that one.
Yes, Naveen, happy to. It's an important question, and we actually used it, the 50% cutoff, in two different settings. So the first one, and Katie showed one slide on it. So the first one was for the binary or the two-level variable. And there we combined the data across all cohorts. And in this case, the analysis did not take into account the different ranges of BAF values for the different assays. But what we did, we used an optimal cut point analysis in overall survival to determine this cut point. And this analysis, it actually looks at the hazard ratio at each, and does a log rank test at each possible cut point. And then we maximized the hazard ratio, and the optimal cut point found was that it was a 50% decrease in ctDNA, which maximized the difference in overall survival. And then patients were assigned to the two groups having a large decrease that exceeded the 50%, and all the others. So that was for the binary level. And in contrast, we also looked at the 3-level variable, and that was nice because we could do a cohort-specific category, unlike, across all cohorts. And for this one, the 50% means something very different. For this one, we did within each cohort, we ordered patients based on their ctDNA % change values. And then among all the patients with a decrease, we took a 50% cut point and took the 50% of the most decrease we called true decrease or extreme decrease. And then we did the same for the increase. Everybody with a 50% increase, everybody with an increase was cut into the 50% most extreme increases. And then the two most extreme groups were the increase and decrease, and everybody in between was the intermediate group. And importantly, both of these variables gave, or both metrics were similarly associated with all the outcomes. So independent of whether we looked at the 3-level cohort specific or the 2-level across cohorts.
Thank you, Antje. So, to summarize, it looks like if you apply a universal cut point, which is at the 50% point, or whether you provide a cohort specific, so kind of a reference-based approach, 50% results are very consistent across the two. But obviously the question of thresholds is one that is going to come up quite a bit in step 2 as we look at different settings and different cancer types. So let's bear that in mind. There's a question that's come up just now on the completeness of the data. And let's start maybe with a question that is, in terms of the data set that we had, the 200 patients, how complete were the baselines, and what did we do with patients who might have had non-detectable ctDNA at baseline? And I'm going to go back to the statisticians, and Chi, you were at the table for this one, so maybe you could answer it.
Sure. Actually, there are about 6% of patients who had no detectable ctDNA at baseline. But then you would wonder, well, are they excluded from the analysis? But no, we did not exclude them because they are not missing data points. So basically, a patient did not have detectable ctDNA either because they did not have tumor cells that's shedding ctDNA, or there were tumor cells, but our assays are not sensitive enough to pick up the signal. So in either case, excluding them from the analysis would be informative sensory. So instead of excluding them, what we did is we set the value to 0. It's not perfect, but we think that's the reasonable approach given the current data.
Thank you, Chi. So I want to go back to this question of the variance and the types of assays. So first, let's refresh our memory. How many assays did we have in step 1? And Diana, maybe you could tell us the answer to that one.
Sure, that sounds great. So as you saw from the presentation we had 2 weeks ago and the review that Katie went through over this earlier today, we had 4 different diagnostic companies represented in the data that we received. And that's with a total of 5 assays, and I have them here. So we had the Confera assay for Langston Bio, we had the Guardant 360, the Guardant Omni, the Roche Avenio, Hopkins used the TechSeq NGS. And what we, obviously we didn't expect that to have these many assays, but I think that reflects what is going on in the field right now, and that there are many different assays that are available to do this type of work. And that is something that we did not restrict as we asked for the data. That didn't mean that it wasn't a challenge to try to harmonize across of them, but there were some similarities that we could use to assess the data in a similar way across all those different assays.
Good. And there's another question that's come up, and I'd be curious, John, on your views on that, and maybe some of the others. There's a very specific question that's come up on, so given that we have this different approach from the different assays, what are the observations, what are the thoughts on how we capture tumor load?
So yeah, I mean, obviously when we look at ctDNA metrics, and it's going to be limited to the prescribed assay for the ctDNA analysis itself. So we're looking at the analytical characteristics of those assays and the variants targeted. And each of these 4 different assays that were utilized within step 1, obviously they have different characteristics. And that's, dependent on the sensitivity of those assays and the limitations of those assays, going to be able to determine, how sensitive we can be able to detect for the ctDNA variants that we're targeting. And also the methodology that's used within those varying assays as well. I mean, some are targeted panels for whole genome, but the sulfite sequencing methodologies. So those are all going to influence in how we detect the overall tumor burden.
Any other thoughts on this question of signal to noise and even the volumes of the samples or the clinical setting? Any other thoughts from the panel? I see Jeff's hand is up.
One of the things that many assays are doing is they're normalizing the tumor signal to the overall signal in the DNA. And so it's a fraction, a tumor fraction or a ctDNA fraction or an allelic fraction, right? And that is a measure that is actually relatively easily scaled from assay to assay. And so an AF on a digital PCR assay is comparable to an AF on an NGS assay. The question, I think, when I read it was that, what if the sort of white cell volume changes? The patient has an inflammatory process, and so their overall DNA goes up, but their tumor DNA stays the same. You would see an apparent dilution of the tumor signal, and so the tumor fraction could go down due to a change in the overall DNA and not a change in the tumor DNA. And so this question is, what's the best way to measure the overall tumor DNA? Should you measure the tumor DNA in and of itself within the volume measured, or the fraction of DNA? And the short answer is we don't know the answer to that. It's an interesting question, but measuring the fraction is certainly a much more scalable and flexible measure, which is why it's been used across so many more assays. It's relatively more difficult to measure the exact DNA content in a blood specimen without that fraction assessment.
So it sounds like this might be an important point. So Rena, I might come to you in a minute, but first I think Darya, you wanted to chime in with some commentary.
Yep. I think the goal of step 1 was to characterize biology of the process, right? Do ctDNA levels and shedding levels truly correlate with the outcome variables we're looking for? And do we have technologies that are capable to measure it in order to reflect that? And I think the ctDNA max VAF as a metric used for most of these assays has been shown in this analysis to kind of carry that burden of biology through technology of how you're measuring it to the outcome, and that's the main observation here. As in any diagnostic development, these metrics are going to be evolving over time, and the more data we collect and the more patients we are able to correlate this with, the better the measurements will become. I'm with Jeff on some of the complexities of how to measure it, but I think first order measure is definitely there based on what Katie was showing in the data presentations, and the next steps will continue to refine it, especially as we look to assays that expand beyond ctDNA variant allele frequency as the measurement, and we come up with new metrics to characterize it. But I think that definitely the first order is there to build on.
Good. Thank you for that, Darya. And I wonder, Rena, if you might have some commentary to add. I'm sure you see this all the time in your work at CDRH. You might need to unmute, Rena. There you go.
Sorry, I was actually using a cell phone, so, okay. So multiple sponsors actually look into different ways of calculating the ctDNA. I think some Jeff and Darya pointed out. So, you know, in this study, it's looking at the variants, the VAF, but we're also seeing patient-specific panels. That's another way of looking at monitoring the response to treatment. Probably Jeff and Darya can comment on that. And with regards to the matrix, it's actually a very important question to understand what is the right metric to look at, for example, for the limit of detection of the assay, whether it's the tumor fraction or whether, you know, there are some other metrics that could be the right metric for the limit of detection of the assay. And, you know, I want to emphasize on, I guess this is the area the community needs to standardize and harmonize in a going forward, or probably in step 2.
Naveen, I believe you're on mute now.
Thank you. Sorry about that. So it's very clear we have some good marching orders of at least one set of questions we have to answer in step 2 as a community. Thank you for that, Rena. There's a question that's come up, and Antje, I think it might be for you, possibly for Chi, and it has to do with, we have shown the outcome assessment based on change in max VAF. And the question is, did we assess change in mean or median VAF? And if so, what were the results? So, Antje.
Yeah, we assessed, so all the analysis we showed you were for max VAF, but we did them all also for mean and median VAF. And the signal had consistently be strongest for max VAF, but the other signals were just a little weaker. But we got pretty consistent results.
Okay, very good. Chi, anything to add?
Yeah, so I agree with what Antje said, and we did look into different different methods. I guess, as we all know, that all models are wrong, except some makes more sense than others. So I already heard questions like, why do you do 2-level, 3-level, not other levels? Or, you know, you use max and you didn't do a lot in the mean, median, all that. I guess the answer, well, we tried a lot of things. We only presented a small fraction of the analysis we did. And then, you know, the CRAB have done a tremendous amount of work over the last few months. At the end of the day, we're happy to see that no matter how you see the data, no matter how you analyze it, the signal is there. It's robust and it's consistent.
Okay, there you go. So definitely worth further study on that basis. So I want to go to a question that we've heard previously. And I don't know, Carl, if you'll be comfortable responding to this one. But it has to do with, well, first there's a question that we have to clarify, which is what was the time window during which we had samples in step 1? So what was the baseline time window and what was the post-baseline time window? And Antje, I think I'm going to send this one to you again.
Yeah, so the baseline time window was within 14 days before start of therapy. And the first time point after initiation of therapy had to be within 70 days. And, you know, we had a lot of discussions as to what window to use for both of them. And we really wanted to be inclusive and really make sure that we capture as much data as possible. We would have preferred a little earlier window, but that would have excluded too many patients.
Okay, so thank you for that. So then the question is, in those 70 days, Carl, and others, if you're interested in responding or adding after Carl has spoken, did we look actually at what happens early in those 70 days versus later in the 70 days?
In this particular data set, we were limited, I think, for the 3 of the 4 assays for a single time point post-therapy. It varied as was said, but I think, you know, this question of what's the right time to assay this is really a key question that we have to address in step 2. I did want to comment on the prior discussion about the variant levels, whether it should be absolute or relative. To me, the really interesting thing about this study was the fact that 4 different assays all gave us the same sort of predictability of response. And so to me, that's really interesting and encouraging. But we must keep in mind that these were all with 4 very active drugs in this particular population. So it's certainly not surprising that we did get similar findings with each one of them. As we extend this to other drugs, perhaps ones that are less active or even inactive, obviously the time windows will be critical. Whether we're looking at absolute versus relative levels of the variants will be important. We do know that with some very active drugs like osimertinib, which is an EGFR inhibitor, we can see changes within 5 days. We clearly see better changes at 3 weeks or 6 weeks. But I think understanding the kinetics of the drop of ctDNA will be very enlightening as we try to understand the mechanism of action of different drugs in this setting.
Good. Thank you for that. Any other comments on this point?
I'm happy to echo that 3-week idea. I think that is the exciting time frame as a clinician, right? When you're seeing a patient, you want to be able to say, well, I'll see you in a few weeks and we'll see how you're feeling on the drug and we'll see if it's working. You know what I mean? And if you could send a blood test a couple weeks in and already at that time point anticipate the results of the scan that ends up being 6, 8, 12 weeks down the line, that would be compelling clinically, and I think potentially compelling from a drug development standpoint to be able to get that early signal of activity. Carl mentioned 5 days. I think in some clinical trials, you see a patient in 3 or 5 days. That's not sort of clinically as scalable a solution. To me, it's really that first visit, 1, 2, 3 weeks in, where there's a lot of potential value. We just need to prove it, ideally, in the next phase of the investigation.
I want to bring in Matt and Roy on this and start to shift the conversation to what we do next. So also for the audience, start thinking about what's on your mind, because we're going to go to you in a second, well, probably in 10 minutes. So maybe for the two of you first, is it feasible to consider in usual clinical care to have ctDNA as early as Jeff and Carl have suggested? Matt, maybe you first and then Roy.
Sure. I think I wanted to start echoing something that Darya in particular highlighted, which is that part of these results, as well as others in the field, what they've really highlighted is that ctDNA gives us an opportunity to interpret the in vivo disease state of what's happening in a patient in real time. And that can't be underestimated. I think that is an exceptionally profound use of this technology, which is not a given if we back up to just a few years ago. Between the routine detectability of most patients with metastatic disease, it offers opportunities to build new endpoints in clinical trials, to refine signal to noise ratios in drug development, to inform clinical practice. I think if anything, the applicability of ctDNA as a adjunct to our clinical care might be underappreciated, in part because of some of the technical optimization that's still necessary. But as a tool, I think that we're starting from a point of this is biologically real. So in that way, it is absolutely worth doing the work that's needed to technically optimize and to provide confidence and use cases for which this tool can be used. But I think it's beyond doubt that it can and should be useful. And that's a turning point, I think, in the application of this assay, where I think that there might have been skepticism about detectability and applicability even a short while ago. And so I think that that's worth sort of not losing sight of, just how distinct the opportunity is to examine the biology of what's happening in a patient. And I guess I would extend that to then say that some of the conversations around what's the right time point and so forth set up situations in which we're competing against radiology. Radiology assessments of what's happening have been really important and a bedrock of drug development and clinical practice, of course. But I would argue actually that although some of these settings in which you can get an even earlier signal are particularly important, there's also settings in which radiology is ambiguous at best, and such that the opportunity to integrate ctDNA in situations where radiology is insufficient might actually be a quicker path towards clinical use. And that includes in the adjuvant setting where there's nothing that you can't see something. But patients are, still at high risk of recurrence, which might be seen only microscopically. It's true in the context of radiologic stable disease, where we can't really differentiate whether a patient's benefiting or not. And particularly in the context of immunotherapy, where long-term responders often have radiologically static imaging, but it's unclear whether they have residual cancer, this is a setting in which ctDNA can provide new insight. So I think in addition to sort of competing with or providing earlier assessments of disease, there's already clinical settings in which radiology is ambiguous at best, and ctDNA can provide new insight.
So I think that brings up the question of how we use it as an adjunct to the current tools in the armamentarium. Roy, I wonder if we might go to you for a commentary.
Oh, thank you, and congratulations on the project. Absolutely, I'm sitting in the clinic now, and this is just so patient-friendly and feasible that we've seen a whole number of patients this morning, and we send off this all the time, for as an adjunct to care. So having this as a tool and given the results we've seen today, it has immediate clinical applicability. How often, especially in this era of treating with the immunotherapy, targeted therapy too, we don't know if we have a stable disease or progression, and we want to get a sense for what to do, especially if the patient's feeling well. So this is a very useful tool. I think, in listening to the discussion, and this is my second panel, I think prospective studies are the next step. So with all the issues we've heard about, when to test, what is the right cutoff point, that will be the next step. Prospective studies, incorporating this into trials. I can tell you, even since the panel 2 weeks ago, I've had several calls from collaborators talking about why don't we start to think about including this in our next trial. So that's going to be and there are plenty of those, by the way. So I think that's going to be really useful. But right now, already clinically applicable, agree correlation with radiology when possible. But there are many settings where radiology either can't be done or isn't sensitive enough. So this has a lot of use.
Thank you. I'm thinking of maybe linking two of the conversations we've had. One is around other tumor settings and exactly what would be the contribution of ctDNA. And then this question of limit of detection. And that to me kind of takes us into the adjuvant setting, where our technologies right now may have low ctDNA. And so Erik, I may come to you in a moment for your thoughts on this. And as we're thinking of, let's go ahead and have, think ahead to what our step 2 trial should be, what are the thoughts around the adjuvant setting from the panel? Is it particularly challenging? What do we need to be prepared for if we go there? Thoughts? Is there anyone who wants to pick this up?
I'm going to try to reference a principle with RESIST, which is this historical tool we use for at least drug assessment, right? And with RESIST imaging response assessment, you need measurable disease. You need some baseline level of tumor to measure and to see change. And so one of the reasons why I see advanced disease as low-hanging fruit is that we have such a facile ability to measure our tumor content in advanced disease and therefore measure change. And as we get to lower and lower levels of tumor content, the ability to assess response is more limited. Thus the sort of approach that we use here, where there was I think if I'm referencing right, you said if there was no tumor evident at baseline, then the only result the patient could have was no response, because 0 goes to 0, which is 0 change, right? And so you need to have some baseline level to see that change. I see advanced disease as sort of right in front of us right now. Certainly there's excitement as our technologies improve to get to the adjuvant setting where, you really have nothing to watch, but you're looking at signals that are so low that I think we need a good assessment of what is a response when there's more tumor present, and then we can reel that back to early stage when our reference point is much smaller. That would be my idea.
Okay. And Erik, I wonder if you have thoughts on this. This is now taking what is a pretty reasonable signal in the advanced setting, trying to move it upstream. What advice would you have for us as we consider the adjuvant setting for trial design? Or...
Sure. So, I mean, let's say you borrow some of the adjuvant designs in breast cancer and the large trials there for recurrence. Those trials are very large. You know, they're in the sometimes 4,000 to 5,000 patient range, and it's just sort of an event issue. Sometimes maybe you only have 10% of patients recurring at 3 years. And so, you know, practically speaking, if we want to move to the prospective sort of trials, the metastatic trials typically have smaller sample size here. So I think for just, let's say, a quicker resolution of data and like a more like a feasibility aspect, the metastatic, I think is a better place to start simply for a sample size issue. I think that's typically how drug development occurs. We typically will see in the refractory setting sort of development, and then you move earlier. But I think the adjuvant setting is clearly, you know, a great option if to see, like, say, early recurrence and to possibly treat. But I think the metastatic for feasibility is probably a more appropriate place to begin. And I just echo what Jeff also said.
Thank you, Erik. Other thoughts on the question of settings we might want to study in step 2? Heard a very nice expose from Erik, and we've talked about adjuvant. Any other thoughts there?
You know, there are studies in the literature showing that one can identify patients that are given intent to cure treatment, such as surgery and chemotherapy, where there's a patient population that's still unmet need, and that can be identified by persistence of ctDNA, you know, after a few months after those therapies. To be able to detect that, it's a very low level. If you think about sort of a 1 or 2 centimeter tumor, you really need to get down to a sensitivity of 0.01%. And many of the assays have improved greatly in terms of their specificity, but there aren't too many that can enable you to go that low. Plus, you have to have enough variance within the population to detect the patient that still is shedding DNA. So some of the newer assays looking at tumor-informed bespoke assays where you can look at, you know, 15 to 50 different variants gives you greater sensitivity, and those studies are in progress now.
Okay. I think I'd like to ask the audience now if you could help us with your advice. If you could put into your chat or into the Q&A your views on what we should do next for step 2, where your priorities would be, where your greatest interests are, and if you have data that you want to contribute, please feel free. So if you could start populating the chat box, and what I'd like to do then in the time that remains is give the panel the opportunity to comment for themselves where they see priorities and what advice they have for us as we undertake this work. And so I think I'll start in the order that you were introduced, which is probably the safest way. It's alphabetical. And so John, you get the first go at this.
Yeah, so I mean, the way that I look at it is there's opportunity across the entire paradigm. Obviously, I think Geoffrey noted it well, and so did Erik. I mean, the metastatic space, I mean, there's low-hanging fruit that can be aggressively pursued for ctDNA analysis. But obviously, as we look into the adjuvant phase and early stage disease, where we're looking at minimal residual disease or molecular residual disease or areas of opportunity. Also, you know, there's, you know, even going further and looking at in the neoadjuvant phase as well. So I think, you know, there's opportunities across the whole paradigm that we should explore. Obviously, they're going to be tumor dependent based on the tumor shed rate for those tumor types that should be considered, but there's opportunities that I foresee across.
Thank you, John. Carl, your priorities for step 2.
Yeah, I think we really need to understand, you know, the variation between tumor types and between different classes or drugs involved in this process. And so I think much more data looking at the kinetics, as we said, to look for the depth of response. You know, does it matter whether you're getting, you know, a 50% decrease versus a 100 or 1,000-fold decrease, I would imagine that would be a big difference in terms of the outcome. I think the other area that is important to look at is not only can we identify those patients that are responding faster than we can with imaging, but can we also find those patients that are failing fast, before we detect them through progression by imaging. And there's lots of data to suggest that rises in ctDNA can precede by several months, resist progression. And that, I think, represents an opportunity to provide patients with a new therapy.
Yes, and that ties into a comment we're seeing from the audience around having serial time points post-baseline. And if we're interested in the question that you've posed and providing more evidence around the relationship between the kinetics and then ultimate clinical course, we would want some of those early time points for sure. Yeah, thank you, Carl. Erik, your advice for the group as we embark on phase 2, what should we focus on from your perspective?
Sure. So I just want to, what Carl said about identifying some of the early responders as well as early progressors. I think this marker could be used for both. I think advice is, I think the ultimate hope is something like this could be used to support marketing applications similar to how PFS started to be adopted, let's say 15 years ago. I think the early data presented by Katie was very, suggestive, and I think it was to suggest that there is something going on here. I think what's needed is more, let's say, prospectively studies, and we also need the results from failed studies here. So sometimes we'll see the results of how the ctDNA works with response rate on studies with positive effects. I think we also need those from the failed studies too, ones where the drugs don't work. And what that'll allow you to do is if you're able to see a correlation, if they're able to give the same type of signals on hazard ratios and response rates, that's going to be very useful to sort of evaluate this endpoint. So, I think more prospective studies, including, let's say, a wide range of cancer types, will go a long way towards this endpoint being better, well adopted.
So I think you're giving us a caution around it's always interesting to provide dichotomies within a trial around the patients that respond and don't. You'd like to see some of the negative trials also.
Yeah, I think I'd also like to see, let's say, trials that don't, let's say the drug, let's say the response rate just wasn't better with the new novel therapy. We'd also like to see that with the ctDNA here too. And what that will do is typically, if you can just think, they like a straight line. And with the negative studies, you're able to get a larger cross section, and it might be able to show a correlation there. Obviously, I think the failed studies are less of an interest, but for this research purpose where you're trying to equate two endpoints, I think it would be important to include those two as well as the positive studies. So.
Good. Thank you. That's clear. That's helpful. Thank you. Why don't we go next to Darya? Your wishes. Where does your mind take you for step 2?
Thank you, Naveen. And it's going to be progressively harder for next panelists to comment given the richness of ideas here. I think I always maintain patient focus on what's actionable to a physician and a clinician when they review a diagnostic result. We've had a lot of discussions about how this work can help with drug development and how academic community is excited about sort of some of the early indications here. I would also encourage us to, in step 2, to think about questions that a clinician would ask and would be able to act on as a result of data that we generate and evidence we generate, and try to start answering those questions as well to provide the background that the community needs in order to use this for the patient benefit. So defining clinically relevant actionable points given serial analysis. What does that look like in clinical practice?
Okay, so we're hearing quite a theme around that serial assessment. We've got a new comment from Erik around the positive and negative so that we could do the classic surrogate marker cross study kind of linkage. Is Matt still with us? I think he had to go. Okay. Roy, your thoughts on step 2?
Well, not wanting to be repetitive, but I think that, adding it, prospectively to trials with either immune therapy agents or targeted agents or combinations would be key. One area where I could see a nice match would be with the lung master protocol, the lung map, where we have many trials starting as a single agent experience, but then randomizing after meeting certain statistical criteria. We're already collecting blood, and we have a strong radiology core as well. So that could be a very nice place to look at it. Certainly, I'd love to see this included more in randomized trials. And then I like the idea of moving to earlier disease. And Carl knows very well the problems with MRD and the sensitivity, but it keeps getting better and better. And I can't help but thinking of a trial that's very much in my mind called ADORA, which was, adjuvant therapy with targeted therapy, osimertinib. And, the hazard ratio looks quite good for DFS. And as we wait for survival results, a very nice analysis there could be to look at the minimal residual disease and use this type of assay in patients to understand early progression, why patients progress, what the mutations are perhaps. And so this technology is going to be used throughout oncology.
Very good. Thank you. In the next 5 minutes, I want to make sure we hear from all our panelists, and I'm actually going to close with Diana to try and round things out. But let's go next to Antje.
Thanks. Most everything's been said. A prospective trial with multiple time points, especially early time points that we can all agree upon are important, would be wonderful. And I really like Roy's suggestions of adding this to lung map, because that would allow us a deeper dive into lung cancer, right? Which we already have some experience now with step 1, and we could really look at the different therapies, right? Because lung map does both targeted and IO therapies. And so I really like that idea. That would be an immediate way to go.
Wonderful, thank you. Chi, could I call on you next? What is your desire for step 2?
Well, thanks, Naveen. Actually, I guess going as one of the last people to comment has the benefit of saying that I agree with all above. And there are a lot of interesting things we can explore. And I really look forward to that, especially in the prospective setting. On top of that, I think I'm very interested in early signal detection, as everybody else has commented on. And I'm also very interested in multi-tumor types. Right? So we have seen step 1 is all on non-small cell lung cancer and all treated with PD-PD-L1. So what if, in other treatment modality and also in different tumor types, which are not shedding so much DNA, ctDNA, is that going to be different? And also something relevant is the surrogacy. And to me, that's like the peak of this effort. And then we have seen this nice association established from step 1, where the association between ctDNA change and clinical outcome is sort of established there. But there are still many steps to go before we establish surrogacy. And I think the beauty or the advantage of this kind of effort is that it's cross-industry, cross-academia, and we have input from our regulatory bodies. So if it takes one company probably 10 years of their own studies to establish surrogacy. So hopefully with all this effort together with Friends of Cancer, probably takes like 3 to 5 years. So we can all accelerate this together.
Thank you, Chi. I love that. Jeff, I'll go to you next, and Rena, and then Diana to bring it home. So Jeff.
I want to echo something Erik said, I think, which was these kind of data could be used as part of regulatory submission, right? That I think is such an immediate opportunity we need to capitalize on. Because when I have that conversation with drug developers, what I get back is convince me this is real. And so what step 2 needs to do is convince everyone that this is so real, so universal, so applicable across diseases and therapies that we are ready to send this data to the FDA to accelerate drug development. And so I would really love a little more conversation around that with the FDA, which is what does that roadmap look like, and how do we make sure that the data we generate ends up being convincing to all parties that this is now one of the ways that we accelerate how drugs are pushed forward to patients.
Okay, and Jeff, I don't think we could have planned that better. Rena, your views on everything you've heard and what advice you'd give us in terms of our work in step 2. Oh, you're on mute again.
So step one was great. Can you hear me now? But my advice also is to look into, the appropriate timing of samples, right? That's very important when we are looking into step two. And also whether this is reproducible across the other disease types and also with the other classes of drugs. And once, you can define what constitutes a ctDNA response in considering the different technologies, different cancer types, different treatment types could have differences in how it constitutes, the definition of ctDNA response. And once we have an understanding of that, a prospective clinical trial, like how Erik was saying in last meeting, Julia Beaver was talking about, that's the next step, once we have an understanding of all the other aspects that I just listed.
Wonderful. So I think that's also clear. Other settings, other drug classes, and understanding what the summary measures would be. Is it one and the same? Is it different? Positive trials, negative trials, etc. Thank you. Diana, I am going to turn this over to you to maybe try and organize our thinking in terms of everything that you have heard. Where does your mind go for step two?
Where doesn't it go?
Where doesn't it go?
Yes. In coordinating the ctMoniTR project and coming across all these wonderful questions, I think that at the beginning, we were all wondering whether it was even going to be possible. And I think that step one showed that this was a data-driven approach. So I want to emphasize basically the last slide of what Katie showed on her presentation, that we would need to see the data available in order to be able to see whether it would be possible to answer these questions. They're all relevant, they're all important, but if we don't have the data available, we literally cannot, as I said, we cannot analyze it. So I definitely encourage everybody who has some thoughts and ideas or has specific questions. I know that a lot of what has been going on through the chat and through the live discussion is centered through a couple of key points. I have jotted down a bunch of little stars between multiple time points, assess the time, the timing of the response and the association between the ctDNA metric and tumor response. Early stages, multiple tumor types. There was a question on the chat about how would it be with breast cancer, with various different subgroups within breast cancer. So all of these questions are great. And we rely on you who are collecting the data to make those possibilities and opportunities available to us. So for step two, I'm a big dreamer. I'm a big optimist. I'd love to be able to have perhaps multiple branches in that if we have a lot of breast cancer data, we could potentially do something specific to breast cancer or something specific to looking at earlier stage of disease. We've talked about IO therapies, perhaps other drug types. That would also be informative. So I think the pie, my wish, I guess, would be, let's see what data is available, what we can do with it, and let's try to answer as many questions as we can with the goal of impacting patients the best or the most. So I want to thank everybody for your time and happy to chat with anybody who'd like to just contact us to be involved in step two.
Very good. So it's all about data. And from there, we will structure our thinking, make sure that we act as methodically in step two as we did in step one, using all the bright minds and all the experts available to us. So a very big thank you to the panel. And now over to Jeff to close out the meeting. Jeff?
Great, thanks so much, Naveen, and thanks to all the participants in today's roundtable discussion. Wow, just a great amount of information, wonderful ideas. Thanks to all of you who attended and participated through the chat and have followed up even since our meeting on the 11th. We really look forward to building out step two into the collaboration that really has been driving ctMoniTR step one. We're really proud that this work has stayed on track despite the adverse circumstances that everyone's been facing. And that's a true testament to the teams involved. So thank you all very, very much. As a next step, we'll be following up with numerous interested parties to explore how we can expand on these efforts into step 2. And this would include looking at different treatment settings and drug classes, as you've heard our experts talk about today. As we begin to align the logistics and assess the feasibility for additional modules to complement the initial work in IO-treated lung cancer, we hope that many of your organizations will consider being a part of this team and help us build ctMoniTR Step 2. So please keep an eye out on a different topic for our next virtual meeting, which is coming in 2 parts on September 21st and 22nd. This is going to be more like our past virtual meetings and include the results presentations from our real-world evidence pilot project 2.0. Until then, thank you all for your continued support of Friends, and please take care and be safe. Thanks.